Hello, and welcome to another chapter of Advanced Pathophysiology. Today we're talking about Chapter 28, the structure and function of the hematologic system. So we will jump right on into it. Again, my pages, as always, may not be lining up perfectly with yours, but it's going to be the entirety of the chapter. Also, if I'm ever going too quickly, you're always welcome to please hit pause. Alright. All the body's tissues and organs require oxygen and nutrients to survive. These essential needs are provided by the blood that flow through miles of vessels through the human body. The red blood cell provides the oxygen and removes carbon dioxide, and the fluid portion of the blood carries the nutrients and ions for proper acid-base balance. The blood also cleans discarded waste from the tissues, transports hormones, conveys white blood cells, platelets, and other ingredients that are necessary for protecting the entire body from injury and infection, and initiating healing, and provides thermal regulation to maintain organ and tissue within an acceptable range of temperatures. So, components of the hematologic system. The composition of blood. Blood consists of various cells that circulate in the cardiovascular system suspended in a solution of protein and inorganic materials, which is approximately 92% water and 8% dissolved substances. The blood volume amounts to about 6 quarts, or 5.5 liters in adults. The continuous movement of blood guarantees that critical components are available to all parts of the body to carry out their chief function: delivery of substances needed for cellular metabolism in the tissues, removal of the waste of cellular metabolism, defense against invading microorganisms and injury, and maintenance of acid-base balance. Fetal and neonatal hematopoiesis and blood composition are going to be discussed in Chapter 30. So, don't you worry, I'll cover that chapter as well. So, talking about plasma and plasma proteins. And actually, before we jump into that, here is the composition of whole blood. It's a good spot to hit pause if you need to review what portion is in proteins versus water, other solutes, and. So, talking about plasma and plasma proteins. In adults, plasma accounts for, again, 50 to 55% of blood volume. Plasma is a complex aqueous liquid containing a variety of organic and inorganic elements. The concentration of these elements varies depending on the diet, metabolic demand, hormones, and vitamins. Plasma differs from serum in that serum is plasma that has been allowed to clot in the laboratory in order to remove fibrinogen and other clotting factors that may interfere with some diagnostic tables. So, this following table here has the constituent and amount of concentration and its major functions. A great spot to hit pause and review all the different components. Let's see if I can get it to where most of it is all in one spot. And again, if I'm ever, excuse me, if I'm ever going too quickly, please hit pause. The plasma contains a large number of proteins, or plasma proteins, that constitute about 7% of the total plasma weight. These vary in structure and function and can be classified into two major groups: albumin and globulins. Most plasma proteins are produced by the liver. The major exception is antibodies, which are produced by plasma cells in the lymph nodes and other lymphoid tissues that were discussed in Chapter 8. Albumin serves as a carrier molecule for normal components of blood as well as drugs that have low solubility in water. It is essential role in its regulation of the passage of water and solutes through the capillaries. Albumin molecules are large and do not diffuse freely through the vascular endothelium. and thus they maintain the critical colloid osmotic pressure that regulates the passage of water and solids into the surrounding tissues. Water and solute particles tend to diffuse out of the arterial portions of the capillaries because the blood pressure is greater in arterial than in venous blood vessels. Water and solutes move from tissue cells into the venous portion of the capillaries, where the pressures are reversed, oncotic pressure being greater than intravascular pressure, or hydrostatic pressure. In the case of decreased production of albumin, or excessive loss of albumin, the reduced oncotic pressure leads to excessive movement of fluids and solutes into the tissue and decreased blood volume. The remaining plasma proteins or globulins are often classified by their properties in an electric field. Under the normal conditions used to perform serum electrophoresis, albumin is most rapidly moving protein. The globulins are classified by their movement relative to albumin. So you have your alpha globulins, the beta globulins, and your gamma globulins. Depending on the electrophoretic procedure, the alpha and beta globulins may be subdivided into subregions, which is why we have our alpha 1 and alpha 2, beta 1 or beta 2 globulins. Fibrinogen is a major plasma protein that would move between the beta and gamma regions, but is removed during the formation of serum. Your gamma globulin region consists primarily of immunoglobulin G. Plasma proteins can be classified into groups by function: clotting, defense, transport, or regulation. The clotting factors promote coagulation and stop bleeding from damaged blood vessels. Fibrinogen is the most plentiful of the clotting factors and is the precursor of the fibrin clot. Proteins involved in defense or protection against infection include antibodies and complement proteins. Transport proteins specifically bind and carry a variety of inorganic and organic molecules, including iron, copper, steroid hormones, and vitamins. The plasma lipids, triglycerides, phospholipids, cholesterol, and fatty acids are carried through the blood as complex or complexes with plasma proteins. They are known as lipoproteins. Regulatory proteins include a variety of enzymatic inhibitors that protect the tissues from damage, precursor molecules that are converted into active biologic molecules when needed, and protein hormones that communicate between the cells. Plasma also contains several charged inorganic ions that regulate cell function, osmotic pressure, and blood pH. So, cellular components of the blood. The cellular elements of the blood are broadly classified as red blood cells, white blood cells, and platelets. The components of the blood are listed in the following table. So here's a good spot to hit pause so you can know the structural characteristic, normal amounts, and function, as well as their lifespan. I'll see if I can get it all into one spot, or as best as possible. So, talking specifically about the erythrocytes, also known as our red blood cells. They are the most abundant cells of the blood, occupying about 48% of the blood volume in men and 42% in women. Erythrocytes are primarily responsible for tissue oxygenation. The erythrocyte contains hemoglobin, which carries the gases and electrolytes, which regulate diffusion through the cell's plasma membrane. The mature erythrocyte lacks a nucleus and cytoplasmic organelles, so it cannot synthesize proteins or carry out oxidative reactions. Because it cannot undergo these mitotic divisions, the erythrocyte has a limited lifespan, usually about 100 to 120 days, and ages, and is removed from the circulation primarily in the spleen to be replaced by new erythrocytes. The erythrocyte's size and shape are ideally suited for its function as a gas carrier. A red blood cell is a small disc with unique properties. a biconcave shape, and a capacity to be reversibly deformed. The flattened biconcave shape provides a surface area-volume ratio that is optimal for gas diffusion into and out of the cell and for deformity. During its lifespan, the erythrocyte, which is 6 to 8 UM in diameter, repeatedly circulates through splenic sinusoids and capillaries that are only 2 UM in diameter. Reversibly, deformity enables the erythrocyte to assume a more compact, torpedo-like shape, squeezes through the microcirculation, and return to its normal shape. So here's a picture of a blood cell. And here you see red cells in the spleen. So again, moving on up to leukocytes, or white blood cell counts, defend the body against microorganisms that cause infection and remove debris, including dead or injured cells of all kinds. The leukocyte acts primarily in the tissue but are transported in the circulation. The average adult has about 5,000 to 10,000 leukocytes. Leukocytes are classified according to structure as either granulocytes or agranulocytes, and according to function as either phagocytes or immunocytes. Granulocytes are neutrophils, basophils, and eosinophils, and are all phagocytes. Mast cells are also classified as granulocytes. Of the agranulocytes, the monocytes and macrophages are phagocytes, whereas lymphocytes are immunocysts, all of which were discussed between chapters 7 and 8. So here we're taking a look at a leukocyte. That brings us to our granulocytes. Granulocytes have many membrane-bound granules in their cytoplasm. These granules contain enzymes capable of killing microorganisms and catabolizing debris ingested during phagocytosis. The granules also contain powerful biochemical mediators with inflammatory and immune functions. These mediators, along with digestive enzymes, are released from granulocytes in response to specific stimuli. The biochemical mediators have vascular and intercellular effects, and the enzymes participate in the breakdown of debris from sites of infection or injury. Granulocytes are capable of amoeboid movement, which they migrate through vessel walls and then to sites where their action is most needed, which was discussed in Chapter 7. The neutrophil is the most numerous and best understood of the granulocytes. Neutrophils constitute between 57 to 67% of the total leukocyte count in adults. The cytoplasm of neutrophils contains small lysosomal granules and a central nucleus with 2 to 5 distinct lobes. Immature neutrophils are called bands or stabs. Mature neutrophils are called segmented neutrophils because of the characteristic appearance of the nucleus. Neutrophils reach a fully mature state in the bone marrow, and these mature neutrophils are called the marrow neutrophil reserve. Normally, it can take up to 14 days for neutrophils to develop from early precursors, but this process is accelerated by infection and treatment with colony-stimulating factors. Neutrophils are the chief phagocytes of early inflammation. Soon after bacterial invasion or tissue injury, neutrophils migrate out of the capillaries and into the inflamed area, where they ingest and destroy microorganisms and debris and then die in 1 to 2 days. The dissolution of dead neutrophils releases digestive enzymes from their cytoplasmic granules. These enzymes dissolve cellular debris and prepare the site for healing. Eosinophils have large coarse granules and constitute only 1% to 4% of the normal leukocyte count in adults. Like neutrophils, eosinophils are capable of amoeboid movement and phagocytosis. Using a spectrum of pattern recognition receptors, eosinophils ingest antigen-antibody complexes and viruses, and are induced by mast cell chemotactic factors to attack parasites and other pathogens. Eosinophil secondary granules contain toxic chemicals that are highly destructive to parasites and viruses. The eosinophil granules contain a variety of enzymes, such as histamines, that help to control inflammatory processes. Eosinophils process antigen and are also released leukotrienes, prostaglandins, platelet activating factor, and a variety of cytokines. So your interleukins 1, 6, your tumor necrosing factor alpha, and granulocyte macrophagy colony stimulating factor, and chemokines that augment the inflammatory response. So your type 1 hypersensitivity allergic reactions and asthma are characterized by a high number of circulating eosinophils that may be involved in a dual role. The regulation of inflammation and contribution to the destructive inflammatory processes observed in the lungs of a person with asthma. Our basophils, which comprise less than 1% of the leukocytes, contain a cytoplasmic granule that have an abundant mixture of biochemical mediators, including histamine, chemotactic factors, proteolytic enzymes, and anticoagulant. Stimulation of basophils also induces a synthesis of vasoactive lipid molecules and cytokines. Basophils produce interleukin 6, which induces interleukin 10 by T helper 1 cells, and also induces T helper 2 cells that favor the B cell differentiation. Basophils also are a particularly rich source of the cytokine interleukin 4, which preferentially guides B cell differentiation towards plasma cells that secrete IgE. The number of basal cells are often increased at sites of allergic inflammatory reactions and parasitic infections, particularly ectoparasites. IgE receptors on the basophil would induce degranulation at sites of IgE-mediated hypersensitivity reactions and contribute to the local inflammatory response. Mast cells are highly similar to basophils, but are generated from a different set of precursory cells in the bone marrow, from which they migrate in an immature form into tissue. They reside in vascularized connective tissue just beneath body epithelial surfaces, including the submucosal tissues of the GI tract and respiratory tract, and the dermal layer that lies just below the surface of the skin. Mast cells play a central role in inflammation, and their activation and degranulation affect a greater number of cells, including those involved in inflammation and healing, as well as the glandular cells and cells of the immune system. Their activation contributes greatly to the increased permeability of blood vessels and synthesis of mediators, producing smooth muscle contractions. Which brings us to the agranulocytes. The agranulocytes, so the monocytes and macrophages and lymphocytes, differ from the granulocytes in that they contain relatively fewer granules in the cytoplasm. Monocytes and macrophages make up the mononuclear phagocytic system. The mononuclear phagocyte system, or MPS, also known as the reticuloendothelial system or macrophage system, consists of specialized endothelial cells in the bone marrow, spleen, and lymph nodes. Promomonocytes and their precursors in the bone marrow, monocytes in the peripheral blood, and the portion of macrophages that reside in the tissue, remaining there for months or perhaps years. Cells of the MPS ingest and destroy unwanted materials, such as foreign protein particles, circulating immune complexes, microorganisms, debris from dead or injured cells, defective or injured erythrocytes, and dead neutrophils. Recently, the osteoclasts was classified as a true member of the MPS. Macrophages stimulate formation of osteoclasts. Multinucleated macrophage-like cells specialize for the function of lacunar bone resorptions and remodeling, in addition to phagocytosis. The following table lists the various names given to macrophages listed in specific tissues. So here's a good spot to hit pause if you're unfamiliar with the different kinds of mononuclear phagocyte systems. So here it's all in one spot, a good spot to hit pause. Monocytes are the largest normal blood cell and have a horseshoe-shaped nucleus. They are formed and released by the bone marrow into the bloodstream. Monocytes migrate into the variety of tissue and fully mature into tissue macrophages and myeloid dendritic cells. Other monocytes may mature into macrophages in the circulation and migrate out of the vessel in response to infection or inflammation. Macrophages are generally larger and are more active as phagocytes than monocytes. Dendritic cells frequently extend projections into the tissue and assume a neuron-like appearance. The origin and turnover of many of the tissue macrophages are not precisely known. It seems clear that once monocytes leave the circulation, they do not return. They can survive many months or even years. The normal role of macrophages is to remove old and damaged cells and large molecular substances from the blood. Cellular targets of macrophage phagocytosis include circulating senescent or damaged erythrocytes and platelets, dead neutrophils, and cells undergoing apoptosis. Non-cellular targets include antigen-antibody complexes, cellular debris, products of coagulation, and macromolecules. Macrophages remove and kill contaminating microorganisms in the blood and at sites of infection. Macrophages, and particularly dendritic cells, are the major antigen processing and antigen presenting cells that initiate immune responses. Macrophages initiate wound healing and tissue remodeling, and if activated by cytokines from T cells, secrete a large array of biologically active chemicals that, if uncontrolled, result in chronic inflammation and tissue injury. Lymphocytes constitute approximately 25 to 33% of the total leukocyte count and are the primary cells of the immune response. Most lymphocytes transiently circulate in the blood and eventually reside in secondary lymphoid tissues as mature T cells, B cells, or plasma cells. The lymphocytes do not contain any enzyme-filled digestive vacuoles. The lifespan of the lymphocyte can be days, months, or years, depending on the type and subtype. Our natural killer cells, therefore, which resemble large granular lymphocytes, kill some types of tumor cells in vitro and some virus-infected cells without being induced by previous exposure to these antigens. Hence, they are named natural killer cells to differentiate them from the T cytotoxic cells, which are induced by an antigen. Our natural killer cells also have the capacity to activate T cells and phagocytes, and produce a variety of cytokines that can regulate immune responses. The predominant form of natural killer cells develops in the bone marrow and circulations in the blood, where it accounts for 5% to 10% of the circulating lymphoid pool, and is found mainly in the peripheral blood and spleen. Natural killer cells develop independent of a thymus, although some natural killer precursors are found in the thymus and may develop into natural killer T cells that have markers of both natural killer and T cells. That brings us to our platelets. Platelets, or thrombocytes, are not true cells, but irregularly shaped cytoplasmic fragments that are essential for blood clotting and control of bleeding. They are formed in the bone marrow by fragmentation of very large cells known as megakaryocytes, a process that normally takes 7 to 10 days. They lack a nucleus and deoxyribonucleic acid and are incapable of mitotic division. They do, however, contain cytoplasmic granules capable of releasing biochemical mediators when stimulated by injury to a blood vessel. These mediators are generally pro-inflammatory. The alpha granules contain a mixture of coagulation factors, growth and angiogenic factors, and angiogenesis inhibitors. Depending upon the particular stimulus, platelets may selectively release promoters or inhibitors of angiogenesis. Activation also stimulates synthesis of arachidonic acid pathway products. Platelets can assume different shapes with pseudopodia and adhere to collagen fibers in damaged vascular walls, plugging vascular openings to help control bleeding. So here we're taking a look at a picture of platelets or megakaryocytes. And then here we're taking a look at a picture of a colored micrograph of platelets. There are roughly 140,000 to 340,000 platelets circulating in the blood. An additional 1/3 of the body's available platelets are in a reserve pool in the spleen. A platelet circulates for about 8 to 11 days, ages, and is removed by macrophages of the MPS, mostly in the spleen. That brings us to the lymphoid organs. The lymphoid system is closely integrated with the circulatory system. The lymphoid organs, some of which are merely aggregations of lymphoid tissue, are classified as primary or secondary. The primary are the thymus and the bone marrow. The secondary consist of the spleen, lymph nodes, tonsils, and Peyer's patches in the ileum of the small intestine. All of the lymphoid organs link the hematologic and immune system in that they are sites of residence, proliferation, differentiation, or function of lymphocyte and mononucleophagocytes. So let's talk about the spleen. The spleen is the largest of the secondary lymphoid organs. It is a site of fetal hematopoiesis, filters blood-borne antigens, and cleanses the blood through the action of mononuclear phagocytes, initiates immune response to blood-borne microorganisms, destroys the aged erythrocytes, and serves as a reservoir for blood. The spleen is a concave, encapsulated organ that weighs about 150 grams and is about the size of the fist. It is located in the left upper abdominal cavity, curved around a portion of the stomach. Strands of connective tissue extend from the capsule, dividing the spleen into compartments. The compartments contain masses of lymphoid tissue, called splenic pulp, with the white pulp embedded within the red pulp. The spleen is interlaced with many blood vessels, some of which are capable of distending to store blood. Blood that circulates through the spleen comes from the splenic artery, which branches from the descending aorta and re-enters the circulating system through the splenic vein and into the portal vein. So here you're taking a look at the splenic architecture, a good spot to pause if you need to renew or refresh yourself from your anatomy. Arterial blood that enters the spleen first encounters the white splenic pulp, which consists of masses of lymphoid tissue containing macrophages and lymphocytes, primarily those T lymphocytes in proximity to the arterioles. Cellular clumps are formed in the white pulp around the splenic arterioles. The lymphoid follicles consist primarily of the B lymphocytes. These are the chief sites of immune function within the spleen. Here, blood-borne antigens encounter lymphocytes, initiating the immune response and the conversion of lymphoid follicles into germinal centers, where B cells proliferate and differentiate during the humoral immune response. Some of the blood that enters the terminal capillaries continues through the microcirculation and enters highly distensible storage areas called venous sinuses in the red pulp of the spleen. The venous sinuses are capable of storing more than 300 milliliters of blood. Passive dilation of the venous sinuses enables the spleen to increase its storage capacity as needed by the body. Sudden reduction in blood pressure causes the sympathetic nervous system to stimulate constriction of the sinuses, resulting in an expulsion of as much as 200 ml of blood into the venous circulation, which helps restore blood volume and increase the hematocrit by as much as 4%. The endothelial lining of the venous sinuses is discontinuous and therefore extremely permeable, so that blood cells are allowed to exit the circulation. The red pulp contains a system of loosely interconnected resident macrophages that provide the principal site of splenic filtration. Because of the slow circulation in the sinuses, the macrophages easily phagocytize old, damaged, and dead blood cells of all kinds, microorganisms, macromolecules, and particles of debris. Hemoglobin from phagocytosed erythrocytes is catabolized, and the heme is stored in the cytoplasm of the macrophages, or released back into the blood to support hemoglobin synthesis. The macrophages also can remove particulate inclusions containing denatured hemoglobin from erythrocytes without harming the cells themselves. Blood that filters through the red pulp also finds its way into the venous sinuses, and hence into the portal circulation. So here you're taking a look at the splenic sinuses. The spleen is not absolutely necessary for life or for adequate hematologic function. However, splenic absence from any cause has several secondary effects on the body. So, for example, leukocytosis often occurs after a splenectomy, suggesting that the spleen exerts some control over the rate of proliferation of leukocyte stem cells in the bone marrow and their release into the bloodstream. Circulating levels of iron may also decrease, really reflecting the spleen's role in the iron cycle. The immune response to encapsulated bacteria, which is primarily an IgM response, may be severely diminished, resulting in increased susceptibility to disseminating infections and sepsis. Loss of the spleen results in an increase in a morphologically defective blood cell in the circulation, as well as senescent platelets, confirming the spleen's role in removing old or damaged red blood cells, and platelets. Thrombocytosis and thrombosis are complications of a splenectomy and can require anticoagulation therapy for 3 to 6 months. Which brings us to lymph nodes. Structurally, lymph nodes are part of the lymphatic system. Lymphatic vessels collect interstitial fluid from the tissues and transport it as lymph through vessels of increasing size to the thoracic duct, which drains into the superior vena cava and returns the lymph to the circulation. Lymph nodes are distributed throughout the body and provide filtration of the lymph during its journey through the lymphatics. Each lymph node is enclosed in a fibrous capsule, branches of which extend inward to partition the node into several compartments. Reticular fibers of connective tissue divide the compartments into a meshwork throughout the lymph node. The node consists of an outer and inner cortical area and an inner medulla. Lymph enters through the multiple small afferent lymphatic vessels into the subcapsular sinus, just beneath the capsule, drains into the cortical sinuses to the medullary sinuses, from which the lymph is collected, and leaves the node by way of the efferent lymphatic vessel. Blood flows into the lymph nodes through the lymphatic artery, which ends in groups of Postcapillary venules distributed throughout the outer cortex. The blood then is drained through the lymphatic vein. So here you're taking a look at the lymph node architecture. A good spot to hit pause if you need a refresher again for that anatomy. Functionally, lymph nodes are part of the hematologic and immune system and are the primary site for the first encounter between antigen and lymphocytes. Lymphocytes enter the lymph node from the blood through the postcapillary venules by means of diapedesis across the endothelial lining. Your B lymphocytes tend to migrate preferentially to nodes in the cortex and medulla, whereas T lymphocytes predominantly migrate to the paracortex. Macrophages reside in the lymph node, help filter the lymph of debris, foreign substances, and microorganisms, and provide antigen processing functions. The dendritic cells encounter and process antigens and microorganisms in other tissues, enter the lymph node through the afferent lymph vessels, and migrate through the nodes. The reticular network provides adhesive surfaces for trapping large numbers of phagocytes and lymphocytes and facilitating their organization into follicles or primary nodules. The presence of antigen, either removed from the lymph by macrophages or presented on the surface of the dendritic cells, results in the production of secondary nodules containing germinal centers. In the germinal centers, lymphocytes, particularly B cells, respond to antigenic stimulation by undergoing proliferation and further differentiation, including class switch into memory cells and plasma cells. Plasma cells migrate to the medullary cords. The B lymphocyte proliferation in response to a great deal of antigen may result in lymph node enlargement and tenderness. Which brings us to the development of blood cells, also known as hematopoiesis. The typical human requires about 100 billion new cells per day. Blood cell production, termed hematopoiesis, is constantly ongoing, occurring in the liver and spleen of the fetus and only in bone marrow after birth. These cells carry out functions including oxygen transport, immunity, and tissue remodeling. The process of hematopoiesis involves the biochemical stimulation of populations of relatively undifferentiated cells to undergo mitotic division and maturation into mature hematologic cells. Although proliferation and differentiation are usually sequential, certain blood cells proliferate and differentiate simultaneously. Erythrocytes and granulocytes generally differentiate fully before entering the blood, but monocytes and lymphocytes continue to mature in the blood and in secondary lymphatic organs. So here in this table you see the human hematopoietic growth factors. A good spot to hit pause so you can review different factors, their cell origin, and the primary cell that is being stimulated. Hematopoiesis continues throughout life, increasing in response to a need to replenish aged red blood cells or destroy circulating cells, or in response to infection. In general, long-term stimuli such as chronic diseases causes a greater increase in hematopoiesis than any kind of acute conditions, such as a hemorrhage. Various abnormalities in medullary hematopoiesis have been identified and are discussed in Chapter 29. Extramedullary hematopoiesis, blood cell production in tissue other than bone marrow of apparently normal blood cells, has been reported in the spleen, liver, and less frequently lymph nodes, adrenal glands, cartilage, adipose tissue, intrathoracic areas, and kidneys. In adults, however, extramedullary hematopoiesis is usually a sign of disease, occurring in pernicious anemia, sickle cell anemia, thalassemia, hemolytic diseases of the newborn, hereditary spherocytosis, and certain leukemias. So, bone marrow. Bone marrow is confined to the cavities of the bone and is the primary site of residence of hematopoietic stem cells. Adults have two kinds of bone marrow, red marrow and yellow marrow. A large quantity of fat in marrow is responsible for its yellow appearance. Not all bones contain active marrow. In adults, active marrow is found primarily in the flat bones of the pelvis, vertebra, cranium, and mandible, and sternum and the ribs, and in extreme proximal portions of the humerus and femur. Inactive marrow predominates in the cavities of other bones. So here we're taking a look at bone marrow. At least two populations of stem cells are found in bone marrow niches. Hematopoietic stem cells are progenitors of all hematologic cells. Both populations of stem cells undergo continuous proliferation and self-renewal in the microenvironment of bone marrow, so that additional HSCs, or hematopoietic stem cells, can be produced to replace those undergoing differentiation. The mesenchymal stem cells, or MSCs, are stromal cells and have a role in maintaining HSCs. They can differentiate into a variety of cells, including osteoblasts, adipocytes, and chondrocytes. The hematologic compartment of the bone marrow consists of a variety of cellular and molecular microenvironments called niches. Niches simultaneously contain stem cells, precursor cells, and terminally differentiated cells, and provide a coordinated signaling network necessary to regulate the self-renewal, differentiation, and maintenance of HSCs. They protect HSCs by maintaining quiescence, limiting their entry into the cell cycle and preventing exhaustion or errors in the DNA replication. The niches of the bone marrow are commonly described as endosteal and vascular, but this is an oversimplification of the complexity that is emerging with more research. There is more likely an array of niches that harbor the cells and cytokines that support hematopoiesis. Niches also have a role in metastasis related to the microenvironment of dormant disseminated cancer cells. So here you're looking at bone marrow stem cell niches. The cellular composition of niches include osteoclasts, osteoblasts, sinusoidal endothelial cells, fibroblasts, adipocytes, and other stromal cells. Osteoblasts are derived from fibroblasts and are responsible for construction of bone. Osteoclasts are multinucleated cells of monocytic origin that remodel bone by resorption. Both cells are located near the endosteum and can produce cytokines that affect proliferation and maintenance of hematopoietic cells. Megakaryocytes and macrophages promote HSC migration and proliferation in the niches. Adipocytes suppress HSC numbers. The area is innervated through the sympathetic nervous system. Hematopoietic marrow is vascularized by the primary arteries of the bone. Arterial vessels enter the bone, branch near the endosteum into smaller arterioles and capillaries, and drain into venous sinusoids. These sinuses coalesce into a large central sinus in the central cavity. Hematopoietic marrow and fat fill the spaces surrounded by a network of venous sinuses. Newly produced blood cells traverse narrow openings between endothelial cells and the venous sinus wall and thus enter the circulation. Dormant or quiescent HSCs localize near small arterioles of the endosteal region that are ensheathed by parasites, which express high levels of nestin and act together with sympathetic nerves to promote HSC maintenance and retention. The low blood flow of the arterial sinusoid network limits gas exchange, creates a relative hypoxic microenvironment, and maintains quiescence. Active HSC are located near the sinusoids, which are likely more diverse in their influence for self-renewal, proliferation, and differentiation. Hematopoietic niches are affected by direct cell-to-cell signaling and by soluble mediators produced by cells within the niches. Niches contain two specialized MSCs, the CXCL12 abundant sinusoidal reticular cells and periarteriolar and sinusoidal nestin expressing cells. These cells closely interact with HSCs and provide intercellular signaling through several HSC regulatory molecules important for retention, expansion, maintenance, and quiescence of HSCs. These molecules include chemokine ligand 12, stem cell factor, vascular cell adhesion molecule 1, and secretoprotein angiopoietin 1. The CAR cell is the predominant cell in the sinusoids. The sympathetic nervous system regulates HSC mobilization through the circadian release of beta-3 norepinephrine, the adrenergic signaling, which downregulates CXCL12 expression in the bone marrow. Nestin-expressing MSCs express large amounts of protein nestin, and particularly SCF and VCAM1. Transforming growth factor beta also plays an important role in regulating HSC behavior, particularly quiescence and self-renewal. Cellular differentiation. All humans originate from a single cell that has the capacity to proliferate and eventually differentiate into a huge diversity of cells of the human body. After fertilization, the egg divides over a 5-day period to form a hollow ball that implants on the uterus. Until about 3 days after fertilization, each cell is undifferentiated and retains the capacity to differentiate into any cell type. In the 5-day blastocyst, the outer layer cell has undergone differentiation and commitment to become the placenta. Cells of the inner mass, however, continue to have unlimited differentiation potential and can grow into different kinds of tissue: blood, nerves, heart, bone, and so forth. After implantation, cells of the inner cell mass begin differentiation into other cell types. Differentiation is a multi-step process and results in intermediate groups of stem cells with more limited, but still impressive, abilities to differentiate into many different types of cells. So here you're taking a look at the differentiation of hematopoietic cells. It's a good spot to hit pause if you don't know how these cells kind of break down into specific cells. Within the bone marrow, niches. Each type of blood cell originates from hematopoietic stem cells that proliferate and differentiate under the control of a variety of cytokines and growth factors. During this process, some hematopoietic stem cells undergo alternative paths of differentiation into more differentiated stem cells that are committed to a particular line of blood cells. As with all stem cells, the hematopoietic stem cells are self-renewing so that a relatively constant population of stem cells is available. Some hematopoietic stem cells will continue differentiation into hematopoietic progenitor cells. Progenitor cells retain proliferative capacity, but are committed to possible further differentiation into particular types of hematopoietic cells. Lymphoid granulocyte monocytes and megakaryocyte erythroid progenitor cells. In addition to intercellular signaling, events between HSCs and cells in the osteoplastic and vascular niches, several cytokines participate in hematopoiesis, particularly colony-stimulating factors, which stimulate the proliferation of progenitor cells and their progeny and initiate the maturation events necessary to produce fully mature cells. Multiple cell types in the hematopoietic organs, including endothelial cells, fibroblasts, and lymphocytes, produce the necessary CSF. So here in this box, you get to read about the clinical uses of colony stimulating factor. It's a good spot to hit pause if you would like to read more about it. You also can see here the morphologic effects of growth factor. So I'll try to pause in certain areas so you can read the most. Hematopoiesis in the bone marrow occurs in two separate pools, the stem cell pool and the bone marrow pool. The stem cell pool is the product of self-renewal that maintains the number of pluripotent stem cells and partially committed progenitor cells. The bone marrow pool contains cells that are proliferating and maturing in preparation for release into the circulation, and mature cells that are stored for later release into the peripheral blood. The peripheral blood also contains two pools of cells, those in the circulation and those stored around the wall of the blood vessel. The marginating storage pool primarily consists of neutrophils that adhere to the endothelium in vessels where the blood flow is relatively slow. These cells can rapidly move into tissue and mucous membranes when needed. So here you're taking a look at hematopoiesis. Under certain conditions of rapid depletion of the circulating pool, the circulating hematologic cells need to be rapidly replenished. Medullary hematopoiesis can be accelerated by any of the following three mechanisms. The first being conversion of yellow bone marrow, which does not produce blood cells, to hematopoietic red marrow by the action of erythropoietin. Then a faster differentiation of progenitor cells, and finally the faster proliferation of stem cells into progenitor cells. The development of erythrocytes. For almost 100 years, it was believed that erythrocytes developed from lymphocytes that were transformed in the spleen. It was not until the 1850s that the bone marrow was identified as the site of erythropoiesis. Bringing us to erythropoiesis. Erythropoiesis is the development of red blood cells. In the confines of the bone marrow, erythroid progenitor cells proliferate and differentiate into large, nucleated proerythroblasts, which are committed into producing cells of the erythroid series. All stages of erythroid development are referred to as erythron. The proerythroblast, which has ribosomes and can produce protein, differentiates through several intermediate forms of erythroblast, while synthesizing hemoglobin and progressively eliminating most intracellular structures, including the nucleus. Thus, the maturing erythroblast becomes more compact and progressively assumes the shape and characteristics of an erythrocyte. Hemoglobin is readily apparent and increases in quantity as nuclear size shrinks through the basophilic and polychromatophilic stages. The orthochromatic erythroblast, also known as normoblast, is the smallest of the nucleated erythrocyte precursors. So here we're taking a look at the erythrocyte differentiation. The last immature form of an erythroblast is the reticulocyte, which is anucleate and contains a mesh-like network of ribosomal nucleic acid that is visible microscopically after being stained with certain dyes. The reticulocyte contains polyribosomes and mitochondria. The reticulocyte matures into an erythrocyte within 24 to 48 hours. During this period, mitochondria and ribosomes disappear and the cell becomes smaller and more disc-like. These final changes, the erythrocyte loses its capacity for hemoglobin synthesis and oxidative metabolism. Reticulocytes remain in the marrow for about 1 day and are released into the venous sinuses. They continue to mature in the bloodstream and may travel to the spleen for several days of additional maturation. The normal reticulocyte count is 1% of the total red blood cell count. About 1% of the body's circulating erythrocyte mass is normally is generated every 24 hours. Therefore, reticulocyte count is a useful clinical index of erythropoietic activity and indicates whether new red cells are being produced or not. The regulation of erythropoiesis. In healthy individuals, the total volume of circulating erythrocytes remains surprisingly constant. Most steps of erythropoiesis are primarily under the control of a feedback loop involving glycoprotein erythropoietin. In conditions of tissue hypoxia, erythropoietin is secreted by the liver and primarily by the peritubular cells of the kidney. Rising levels of circulating erythropoietin cause a compensatory increase in proliferation and differentiation of proerythroblasts in bone marrow. The density of cellular erythropoietin receptor decreases progressively during erythroid maturation to almost undetectable levels on reticulocytes. The normal steady-state rate of production of approximately 2.5 million erythrocytes per second can increase to 17 million per second. During anemia or under conditions of low oxygen concentration, such as a high-altitude environment or pulmonary disease. Thus, the body responds to reduced oxygenation of blood in two ways. Stimulation of chemoreceptors of the carotid body and aortic arch that signals the brain to increase oxygen intake through increased ventilation, and 2. The stimulation of receptors on the kidney peritubular cells to increase erythropoietic synthesis and release, thus increasing the oxygen carrying capacity of the blood. So here you're taking a look at the role of erythropoietin in the regulation of erythropoiesis. Recombinant human erythropoietin is used in individuals with anemia secondary to decreased erythropoietin production from chronic renal failure. An immediate effect of increased endogenous and or exogenous erythropoietin is an increase in the blood's reticulocyte count, followed by increasing levels of erythrocytes. The most significant side effect associated with the recombinant human erythropoietin is increased blood pressure. Our hemoglobin synthesis, therefore. Hemoglobin, the oxygen-carrying protein of the erythrocyte, constitutes roughly 90% of the cell's dry weight. Hemoglobin-packed blood cells take up oxygen in the lungs and exchange it for carbon dioxide in the tissues. A single erythrocyte can contain as many as 300 hemoglobin molecules. Hemoglobin increases the oxygen-carrying capacity of blood by 100-fold. Each hemoglobin molecule is composed of 2 pairs of polypeptide chains and 4 colorful complexes of iron plus protoporphyrin, responsible for blood's ruby red color and oxygen-carrying capacity. So here you're taking a look at the structure of hemoglobin. Several variants of hemoglobin exist, but they differ only slightly in primary structure based on the use of different polypeptide chains: R alpha, beta, gamma, delta, epsilon, or zeta. Each polypeptide chain contains about 150 amino acids and is arranged in a knotted sausage configuration, shown in the figure above. The chains assemble to form a tetrahedron, containing two pairs of identical chains. Hemoglobin A, the most common type in adults, is composed of 2 alpha and 2 beta polypeptide chains. A normal variant, a fetal hemoglobin, is a complex of 2 alpha and 2 gamma polypeptide chains that bind oxygen with a much greater affinity than the adult hemoglobin. So here you see the structure of normal hemoglobin molecules. A good spot to hit pause if you're unfamiliar with the different types. Heme is a large, flat iron protoporphyrin disk that is synthesized in the mitochondria and carry one molecule of oxygen. Thus, an individual hemoglobin molecule with its 4 hemes can carry 4 oxygen molecules. If all 4 oxygen binding sites are occupied by oxygen, the molecule is said to be saturated. Through a series of complex biochemical reactions, protoporphyrin, a complex 4-ringed molecule, is produced and bound with ferrous iron. It is crucial that the iron be correctly charged. Reduced ferrous iron can bind oxygen in the lungs and release in the tissues, where oxygen concentration is less, whereas ferric iron cannot. Binding of oxygen to ferrous iron temporarily oxidizes your iron to a more positively charged iron. But after the release of oxygen, the body reduces the iron to its deoxyhemoglobin and reactivates the hemoglobin's capacity to bind oxygen. Without the reactivation by the methemoglobin reductase, the iron positive containing hemoglobin cannot bind oxygen. An excess of ferric iron occurs with certain drugs and chemicals, such as our nitrates and sulfonamides, and reduces oxygen carrying capacity. Hemoglobin also undergoes a conformational change when binding oxygen. When one of the iron molecules binds oxygen, the porphyrin ring changes shape, increases the exposure of the 3 remaining iron atoms to oxygen. This greatly increases the affinity of the oxygen-carrying capacity of hemoglobin as it occurs in the lungs. When oxygen is unloaded from hemoglobin, the oxygen-carrying capacity of hemoglobin is low, facilitating the transport of carbon dioxide back to the lungs. Several other molecules can competitively bind to deoxyhemoglobin. Carbon monoxide directly competes with oxygen for binding to ferrous ion with an affinity that is about 200-fold greater than that of oxygen. Thus, even a small amount of CO2 can dramatically decrease the ability of hemoglobin to bind and transport oxygen. Hemoglobin also binds carbon dioxide, but at a binding site separate from where oxygen binds. In the lungs, CO2 is released, allowing hemoglobin to bind to oxygen. Erythrocytes may play a role in the maintenance of vascular relaxation. Nitric oxide produced by blood vessels is a major mediator of relaxation and dilation of the vessel wall. In the lungs, hemoglobin can concurrently bind oxygen to ferrous iron, and nitric oxide to cysteine residues in the globins. As hemoglobin transfers its oxygen to tissue, it may also shed small amounts of nitric oxide, contributing to dilation of the blood vessel and helping the oxygen regain access to tissues. Nutritional requirements for erythropoiesis. Normal development of erythrocytes and synthesis of hemoglobin. depend on an optimal biochemical milieu and adequate supply of the necessary building blocks, including the proteins, vitamins, and minerals. If these components are lacking for a prolonged time, erythrocyte production slows and anemia may result. So here in this following box, we're looking at the nutritional requirements for erythropoiesis. A good spot to pause. And again, as always, I'll chart them, put it all as best as possible into one spot. Erythropoiesis cannot proceed in the absence of vitamins, especially B12, folate, B6, riboflavin, pantothenic acid, niacin, ascorbic acid, and vitamin E. Your dietary vitamin B12 is a large molecule that requires a protein secreted by parietal cells into the stomach for transport across the ileum. Once absorbed, vitamin B12 is stored in the liver and used as needed in erythropoiesis. Defects in IF production lead to decreased B12 absorption and pernicious anemia. Folate is the second most important vitamin for erythrocyte production and maturation. Folate is necessary for DNA synthesis, being a component of 3 of the 4 DNA bases are thymine, adenine, and guanine, and RNA synthesis. Folate absorption occurs principally in the upper small intestine and it is stored in the liver. Folate deficiency is more common than a vitamin B12 deficiency and occurs more rapidly. Folate stores can be depleted within a few months, whereas vitamin B12 depletion can take years. Folate supplements are prescribed for pregnant women because pregnancy increases the demand for folate and may cause anemia. Supplements can protect against neural tube defects and may prevent anemia. So, the normal destruction of senescent erythrocytes. After about 100 to 120 days in the circulation, old erythrocytes are removed by tissue macrophages, primarily in the spleen. Although mature erythrocytes lack nuclei, mitochondria, and the endoplasmic reticulum, they do have cytoplasmic enzymes capable of glycolysis and production of small quantities of ATP, which provides the energy needed to maintain cell function and membrane pliability. Metabolic processes diminish as the erythrocytes ages, so less ATP is available to maintain plasma membrane function. Disruption of the anchorage between the cytoskeleton and the plasma membrane result in a senescent red cell becoming increasingly fragile and losing its reversible deformability, and thus becoming susceptible to rupture while passing through narrowed regions of the microcirculation. Additionally, the plasma membrane of the senescent red cells undergoes a phospholipid rearrangement that is recognized by receptors for the phosphatidylserine on macrophages that selectively remove and sequester the red cell. If the spleen is dysfunctional or absent, macrophages in the liver become responsible for this process via the Kupffer cells. The erythrocytes are digested by proteolytic and lipolytic enzymes in the phagolysosomes of the macrophage. The heme and globin of the methemoglobin dissociate easily, and the globin is broken down into its component amino acids. The iron in hemoglobin is oxidized, forming the methemoglobin and recycled. Phosphorus is reduced to bilirubin, which is transported to the liver, conjugated, and finally excreted in the bile as glucuronide. Approximately 6 grams of hemoglobin is catabolized daily, producing 200 milligrams of bilirubin. Bacteria in the intestinal lumen transform conjugated bilirubin into urobilinogen, Although a small portion is reabsorbed for further metabolism by the liver or excreted by the kidney into the urine, most urobilinogen is excreted in the feces. Conditions causing accelerated erythrocyte destruction increase the load of bilirubin for hepatic clearance, leading to increased serum levels of unconjugated bilirubin and increased urinary excretion of the urobilinogen. Your gallstones or cholelithiasis can result from a chronically elevated rate of bilirubin excretion. So here you're taking a look at the metabolism of bilirubin released by heme breakdown. The iron cycle. Roughly 67% of the total body iron is bound to heme in erythrocytes and muscle cells, and roughly 30% is stored in mononuclear phagocytes and hepatic parenchymal cells as either ferritin or hemosiderin. The remaining 3% is lost daily by expelling urine, sweat, and bile, sloughing epithelial cells from the skin and intestinal mucosa, and minor bleeding. So, roughly 25 milligrams of iron is required daily for erythropoiesis. Only 1 to 2 milligrams of iron is dietary, and the remainder is absorbed from iron recycling of prior erythrocytes. The methemoglobin released from the breakdown of senescent or damaged erythrocytes is disassociated by the enzyme heme oxygenase, and iron is released into the bloodstream, where it is free to bind again to transferrin or be stored in the macrophage cytoplasm as ferritin or hemosiderin. A minute amount of iron or a minute amount of iron is stored in the muscle cells by the heme-containing protein myoglobin. Unavailable stores of iron are present in cytochromes, catalases, and peroxidase enzymes. So here you're taking a look at the iron cycle. The protein ferritin is the major intracellular iron storage protein. Apoferritin, which is ferritin without attached iron, can store thousands of atoms of iron. Several apoferritin complexes combine to form the micelle ferritin. Large aggregates of micelles produce numerous ferritin micelles, known as hemosiderin. Hemosiderin is visible as an iron-based pigment under the light microscope as cell inclusions. The iron within deposits of hemosiderin is poorly available to supply iron as needed. The most common cause of hemosiderin deposition is simple bruising. Hemosiderin in small amounts within iron-rich tissue is considered normal. Large aggregates, or its presence in tissue such as the lungs or subcutaneous tissue, suggests a pathologic condition. Iron, acquired by utilization of dietary sources, release of iron stores, or catabolism of erythrocytes is transported in the blood bound to apotransferrin, thus becoming transferrin. Under normal conditions, only 1/3 of the iron binding sites on transferrin molecules are occupied. Apotransferrin is a glycoprotein synthesized primarily by hepatocytes in the liver, but also produced in small quantities by tissue macrophages, submaxillary and mammillary glands, and ovaries or testes. Iron for hemoglobin production is carried by transferrin to the bone marrow, where it binds to transferrin receptors on erythroblasts. Transferrin receptors are on the plasma membrane of all nucleated cells. Although they are at particularly high levels of erythroid precursors and rapidly proliferating cells, and are thought to be the only route of cellular entry for transferrin-attached iron. Transferrin is recycled by intracellular dissociation of the iron with secretion of the resultant apotransferrin into the bloodstream. The iron is transported to the erythroblast mitochondria, where the enzyme heme synthesase inserts ferrous iron into protoporphyrin to form heme. Heme is bound to globin to form hemoglobin. Iron not used in erythropoiesis is stored temporarily as ferritin, or hemosiderin, and later excreted. Splenic red pulp macrophages are specialized for iron recycling with increased expression of proteins for the uptake of hemoglobin, the breakdown of heme, and the export of iron. The body's iron homeostasis is primarily controlled by the hormone hepcidin. Hepcidin is synthesized in the liver and released as a 25 amino acid peptide, most of which is bound in the plasma, with high affinity to alpha-2 macroglobulin and relatively lower affinity to albumin. Hepatocellular hepcidin production is regulated physiologically by the body's dietary absorption of iron, rate of erythropoiesis, and level of oxygen saturation. Hepatocytes sense levels of circulating iron by means of receptors for transferrin, the major transporter of iron in the plasma. Excess iron is stored in hepatocytes and macrophages. Hepatocytes sense the levels by means of bone morphogenic protein, most likely BMP6, which is a growth factor produced to a large extent by bone marrow sinusoid endothelial cells and the mother against the decapentaplegic protein pathway. Hepcidin production also can be induced by inflammation through the interleukin-6 pathway. Hepcidin regulates iron levels through its binding capacity to ferroportin, which is a transmembrane iron exporter found in the plasma membrane of cells that transport or store iron, including macrophages, hepatocytes, and enterocytes. The body's total iron balance is maintained through controlled absorption rather than excretion. Dietary iron is transported directly across the membranes of epithelial cells in the duodenum and proximal jejunum. Hepcidin induces internalization and degradation of ferroportin, thus leading to increased intracellular iron stores, decreased dietary iron absorption, and decreased levels of circulating iron. Decreased production of hepcidin leads to release of stored iron and increased dietary absorption. Thus, if the body's iron stores are low, or the demand of erythropoiesis increases, dietary iron is transported rapidly through the epithelial cell and into the plasma. If body stores are high and erythropoiesis is not increased, iron transport is stopped, although iron can cross the plasma membranes of epithelial cells and be stored as ferritin. Leading us to the development of leukocytes. Leukocytes consist of lymphocytes, granulocytes, and monocytes. Most of the leukocytes arise from stem cells in the bone marrow. Hematopoietic stem cells differentiate into two populations of progenitor cells, common lymphoid progenitors and common myeloid progenitors. Lymphoid progenitors that remain in the bone marrow undergo differentiation into the B cell lineage, after which they are released into the circulation and undergo further maturation in the peripheral lymphoid organs. The most common myeloid progenitors further differentiate into progenitors for basophils, mast cells, eosinophils, and megakaryocytes, and into the granulocyte monocyte progenitors. The granulocyte monocyte progenitors further differentiate into monocyte progenitors and granulocyte progenitors, which develop into monocyte macrophages and neutrophils, respectively. Development from the hematopoietic stem cell to common granulocyte monocyte progenitors primarily is under the control of the stem cell factor, interleukin 3, and GM-CSF, whereas further differentiation into granulocytic and monocytic progenitors is controlled by the G-CSF and M-CSF, respectively. Monocytic progenitors undergo development into monocytes within 24 hours and are released into the circulation. Monocytes mature within various forms of macrophages, a process that is usually complete in 1 to 2 days after their release. Progenitor cells for granulocytes normally mature in the bone marrow into neutrophils, eosinophils, and basophils. The ultimate phenotype is determined by relative local bone marrow concentrations of early and late acting cytokines, including the GM-CSF, G-CSF, interleukin 3 and 5, stem cell factors, and other portions. Granulocytes are released into the blood within 14 days of development. The bone marrow selectively retains immature granulocytes as a reserve pool that can be rapidly mobilized in response to the body's need. Most leukocytes exist in the body from days to years, depending on the type. Maintenance of optimal levels of granulocytes and monocytes in the blood depend on the availability of pluripotent stem cells in the marrow. Induction of these into committed stem cells, timely release of new cells from the marrow, and mobilization of the granulocyte reserve pool. Leukocyte production increases in response to infection, to the presence of steroids, and to reduction or depletion of reserves in the marrow. It is also associated with strenuous exercise, convulsive seizures, heat, intense radiation, paroxysmal tachycardias, pain, nausea, vomiting, and anxiety. The development of platelets, therefore. Platelets, or thrombocytes, are derived from stem cells and progenitor cells that differentiate into megakaryocytes. During thrombopoiesis, the megakaryocyte progenitor is programmed to undergo an endomitotic cell cycle, during which DNA replication occurs. But anaphase and cytokinesis are blocked. Thus, the megakaryocyte nucleus enlarges and becomes extremely polyploidy without cellular division. Concurrently, the number of cytoplasmic organelles increase and the cell develops the cell surface, proplatelet elongations into the sinusoidal blood vessels. These branches progressively fragment into platelets. A single large megakaryocyte may produce thousands of smaller platelets. Like erythrocytes, platelets released from the bone marrow lack nuclei. About 2/3 of these platelets enter the circulation and the remainder reside in the splenic pool. Platelets circulate in the bloodstream for about 8 to 10 days before losing their ability to perform thrombogenic activity and biochemical reactions. Senescent platelets are sequestered and destroyed in the spleen by mononuclear cell phagocytosis. An adequate level of committed platelet precursors in bone marrow and differentiation into circulating platelets are controlled by specific interactions between the megakaryocyte progenitors and stromal cells in the bone marrow, as well as thrombopoietin and various cytokines and colony-stimulating factors and interleukins. Platelets express high affinity receptors for TPO, and when circulating platelet levels are normal, TPO is absorbed into the platelet surface and prevented from accessing the bone marrow and initiating further platelet production. TPO stimulates committed cells at further stages of differentiation to differentiate faster so that rates of megakaryocyte development, endomitosis, and platelet release are increased. During inflammation, interleukin 6 induces increased synthesis of TPO, which increases production of newly formed platelets, which are more thrombogenic. So, that brings us to the mechanisms of homeostasis. Homeostasis is the arrest of bleeding by the formation of blood clots at sites of vascular injury. As a result of homeostasis, damaged blood vessels maintain a relatively steady state of blood volume, pressure, and flow. 3 equally important interactive components of homeostasis are the vasculature, platelets, and blood proteins. The general sequence of events in homeostasis is the following. You have a vascular injury that leads to a transient arteriolar vasoconstriction to limit blood flow to the affected site. Then you have damage to the endothelial lining or cell lining of the vessel, which exposes the prothrombogenic subendothelial connective tissue matrix, leading to platelet adherence and activation and formation of the hemostatic plug to prevent further bleeding. Then, tissue factors produced by the endothelium collaborate with secreted platelet factors and activated platelets to activate the clotting system to form the fibrin clots and further prevent bleeding. And then the last portion, the fibrin platelet clot, contracts to form a more permanent plug, and regulatory pathways are activated to limit the size of the plug and begin the healing process. The relative importance of the homeostatic mechanisms clearly varies by the vessel size. Damage to large vessels cannot easily be controlled by homeostasis, but requires vascular contraction and dramatically decreases blood flow into the damaged vessel. So here you're taking a look at homeostasis. So the function of blood vessels. The vessel wall consists of a layer of endothelial cells that adhere to an underlying matrix of connective tissue. The matrix contains a variety of proteins, including collagen, fibronectin, and laminins. Endothelial cells adhere to the matrix and to each other through receptors that are expressed only on the intercellular and basal surface. Under normal conditions, the endothelium actively regulates blood flow and prevents spontaneous activation of platelets and the clotting system. Endothelial cells produce nitric oxide from L-arginine and synthesize prostacyclin from arachidonic acid. Both your nitric oxide through the cGMP and PGI2 are vasodilators that work in concert with endothelin to modulate blood flow and pressure via vasoconstriction. Your nitric oxide and your prostaglandin also inhibit platelet adhesion and aggregation. Synergism between prostaglandin and nitric oxide is significant. Prostaglandin production varies a great deal in response to stimuli, whereas nitric oxide is released continually to regulate vascular tone. Nitric oxide has other biologic functions including cell signaling, free radical production, and possible other functions. Endothelium also produces adenosine diphosphatase, which regulates adenosine diphosphatase. The endothelial cell surface contains antithrombotic molecules, such as glycosaminoglycans, thrombomodulin, and plasminogen activators. These limit platelet activation and fibrin deposition. Although thrombomodulin and plasminogen activators help control homeostasis in normal vessels, their effects are magnified during vascular damage and clot formation. Therefore, further information is provided on these molecules in the section of control of homeostatic mechanism. As a result of damage to the vessel, the endothelial cell barrier is frequently compromised. The remaining endothelial cells are activated by products of tissue damage, and the underlying subendothelial matrix is exposed. Endothelial cells contain intracellular structures that contain von Willebrand factor that is released during vascular injury and activates platelets. So the function of platelets. Platelets normally circulate freely, suspended in plasma, in an unactivated state. The roles of platelets are to contribute to regulation of blood flow into the damaged site by induction of vasoconstriction. They also initiate platelet-to-platelet interactions, resulting in formation of the platelet plug to stop further bleeding. And they all activate the coagulation cascade to stabilize the platelet plug. and initiate repair processes, including clot retraction and clot dissolution. The normal platelet count ranges anywhere from 140,000 to 340,000. Thrombocytopenia develops if the platelet count drops below 100,000. The individual may experience prolongation of normal clotting, but is usually not at risk for spontaneous major bleeding episodes, unless the platelet count falls below 20,000. If platelet numbers are elevated, the risk for spontaneous blood clots, stroke, and heart attack is increased. The state of platelet activation is primarily under the control of the endothelial cell lining the wall of the vessels. Damage to the vessel initiates the process of platelet activation. Increased platelet adhesion to the damaged vascular wall, activation that leads to platelet degranulation, which stimulates changes in platelet shape and biochemistry. Aggregation as platelet vascular wall and platelet to platelet adherence increases, and activation of the clotting system, and development of an immobilization meshwork of platelets and fibrin. So here you're taking a look at the platelet activation. Adhesion. Normally, platelets are generally observed rolling along the margins of the vessel. At sites of vessel injury, however, platelets become adherent to the site of endothelial damage, where the subendothelial matrix is exposed and endothelial cells have released from Willebrand factor and decreased their antithrombotic activities. Platelet adhesion is mostly mediated by the binding of platelet surface receptors, that glycoprotein 1b, to von Willebrand factor. The von Willebrand factor protein is found in the subendothelial matrix and is released by the endothelial cells and platelets. Platelet adhesion narrows the diameter of the blood vessel, resulting in increasing shear forces that could strip platelets off the vessel surface. However, those same forces induce conformational changes in the von Willebrand factor molecule that result in increased affinity in your GPIB, thus stabilizing the adherent platelet. So here you're taking a look at blood vessel damage, your platelet activation, your blood clot, and the clot dissolution. An excellent spot to hit pause if you need to refresh yourself on how that whole system works. Platelet adhesion is also facilitated by other interactions between platelet receptors and exposed molecules of the subendothelial matrix. For instance, adhesion is increased through binding of the platelet collagen receptor, your GPVI, and the integrin alpha 2 beta 1 to exposed collagen in that matrix. Bringing us to activation. As a result of interaction with the endothelium or the subendothelial matrix, as well as exposure to inflammatory mediators produced by the endothelium and other cells, the platelets are activated. Activation results in the reorganization of platelet cytoskeleton, leading to dynamic changes in platelet shape from smooth spheres to those with spiny projections, and degranulation, and resulting in the release of various potent biochemicals. Platelets contain three types of granules: dense bodies, alpha granules, and lysosomes. The contents of these dense bodies and alpha granules are particularly important in homeostasis. The dense bodies contain ADP, serotonin, and calcium. ADP recruits and activates other platelets through specific receptors. During activation, the platelet plasma membrane experiences several important changes, including becoming ruffled and sticky, undergoing cellular spreading to make tight contacts between neighboring platelets, causing the platelet plug to seal the injured endothelium, and externalizing the phospholipid phosphatidylserine. which provides a matrix for activation of clotting factors. Serotonin is a vasoactive amine that functions like histamine and increases vasodilation and vascular permeability. Calcium is necessary for many of the adhesive interactions, as well as the intercellular signaling mechanism that control platelet activation. Alpha granules contain a mixture of clotting factors, your fibrinogen and factor V, growth and angiogenic factors, and angiogenesis inhibitors. Platelet factor IV also is a heparin-binding protein that enhances clot formation at the site of injury. Depending upon the particular stimulus, platelets may selectively release promoters or inhibitors of the angiogenesis. Many of these mediators either promote or inhibit platelet activity and the eventual process of clot formation. Your PDGF stimulates smooth muscle cells and promotes tissue repair. Platelet lysosomes have a role in the digestion of phagocytic and cytosolic components, similar to that of nucleated cells. Lysosomal contents also may have important extracellular functions, such as supporting receptor cleavage, fibrinolysis, and degradation of the extracellular matrix components, and remodeling of the vasculature. Platelets also initiate production of the prostaglandin derivative, thromboxane A, which counters the effect of prostaglandins production by the endothelial cells. Thromboxane A, or TXA2, causes vasoconstriction and promotes the degranulation of platelets, increases expression of the platelet fibrinogen receptors, and stimulates platelet aggregation, whereas prostaglandins promotes vasodilation and inhibits platelet degranulation. An isoform of cyclooxygenase converts arachidonic acid to thromboxane A. In platelets, aspirin at low doses specifically and irreversibly inactivates your cyclooxygenase, decreasing production of thromboxane A and decreasing platelet activation and aggregation. Daily intake of low doses of aspirin leads to more than 95% of inhibition of thromboxane A in just a few days. Other stimuli of platelet activation include epinephrine, thrombin, and collagen. Thrombin and collagen are particularly strong stimuli. Thrombin cleaves the extracellular domain of G protein-coupled protease-activated receptors, thereby initiating transmembrane signaling. Aggregation. Platelet aggregation is stimulated primarily by the TXA and ADP, which induce functional fibrinogen receptors on the platelet. Your glycoprotein 2B and 3A complex undergoes a conformational change during activation to become a calcium-dependent receptor for fibrinogen. It is a member of the integrin receptor family and binds other matrix proteins. Defects in expression of your GP2B3A complex, also known as your Glanzmann thrombocythemia, results in a failure to aggregate and diminish clotting times. Although that GP2B3A complex is the most abundant fibrinogen receptor on the platelet, receptors for your von Willebrand factor and collagen also contribute to this process. Interplatelet aggregation and clot retraction, which forms the primary homeostatic plug, are facilitated by fibrinogen bridges between receptors on the platelets. The GP2B3A fibrinogen pathway is essential for the formation of a thrombus and as such is an important therapeutic target for blockage by antiplatelet drugs. In addition, fibrin strands within the clot shorten and become denser and stronger, helping the clot to approximate the edges of the injured vessel wall and sealing the site of injury. Contraction of the myosin and actin filaments in the platelet cytoskeleton mediates platelet contraction and fusion of the platelet mass into the secondary homeostatic plug. Contraction expels serum from the fibrin meshwork, resulting in greater packing and increased strength. This process usually begins within a few minutes after a clot has formed, and most of the serum is expelled within 20 to 60 minutes. If a blood vessel injury is minor, primary homeostasis is achieved by the formation of a platelet plug within 3 to 5 minutes of the injury. Platelet plugs seal the many minute ruptures that occur daily in the microcirculation, particularly in capillaries. With too few platelets, numerous small hemorrhagic areas called purpures develop under the skin and throughout the tissue. If primary homeostasis is inadequate to prevent further bleeding, the process proceeds through secondary homeostasis to create a larger complex of more tightly interactive platelets within a matrix created by activation of the clotting system. Function of the clotting factor. A blood clot is a meshwork of protein strands that stabilizes the platelet plug and traps other cells, such as erythrocytes, phagocytes, and microorganisms. The strands are made of fibrin, which is produced by the clotting system. The clotting system was described in Chapter 7 and consists of a family of proteins that circulate in the plasma of the blood in inactive forms. Initiation of the system results in sequential enzymatic activation of multiple members of the system until a fibrin clot is created. So here's an excellent spot to review the coagulation factors and what their primary function is. Based on laboratory evaluation, the clotting system is commonly presented as two pathways of initiation: the extrinsic pathway and the intrinsic pathway. These pathways join in a common pathway with activation of factor X, which proceeds to thrombin, fibrin, and clot formation. These pathways are controlled by the anticoagulant proteins. The in vivo understanding of the clotting pathways has developed from the clinical disorders that result from deficiency of clotting factors. The extrinsic pathway is activated when there is a vascular injury and blood escapes into tissue with activation of tissue factor. It is the normal mechanism of homeostasis and plug breaks in capillaries that occur with activities of daily living. Tissue factor is abundant in vascular, subendothelial, and adventitial cells surrounding larger blood vessels and body organs, particularly the skin, brain, lungs, kidney, and placenta. There also are receptors for TF on activated monocytes. With vessel trauma and bleeding, TF binds with high affinity to circulating activated factor VII, initiating the clotting pathway. TF is a transmembrane protein that exists in an inactive form but is activated into an active enzyme with factors 9 and 10 as substrates. TF mediates a number of changes in disease-related activities, including inflammation, tumor angiogenesis metastasis, cell migration, and clotting associated with atherosclerosis. The extrinsic pathway is evaluated in vitro by adding TF to a sample of plasma and measuring the prothrombin time. So here you're taking a look at the clotting pathways and control proteins. A good spot to hit pause. And then the second section here shows the factor VII alpha signaling through the tissue factor. Factor 12 alpha, the Hageman factor, your prekallikrein or PK, and the high molecular weight kininogen or HK, initiates the intrinsic coagulation pathway through the activation of the downstream substrate factor 11. The terms intrinsic or contact mean that the pathway is activated within the blood vessel by contacting with anionic surfaces, or the substances without the activation of TF. The intrinsic pathway is not a significant pathway for normal homeostasis, but is a pathophysiologic surface defense mechanism against foreign proteins, microbial pathways or pathogens, and artificial materials. Individuals with deficiencies in intrinsic pathway components do not have disruption of homeostasis or prolonged bleeding, with the exception of hemophilia C. Consequently, factor 12 and factor 11 are being investigated for targeted anticoagulant strategies with lower risk for bleeding tendencies than anticoagulants that target thrombin and factor 10 alpha. The activation of the intrinsic pathway has been investigated using in vitro techniques, an example of which is in the laboratory. Negatively charged substances are introduced to activate factor 12 for the diagnostic plasma clotting test known as the activated partial thromboplastin time, or APTT. Factor 12A recruits HK that is complexed with prekallikrein. Factor 12A activates PK2 kallikrein, which in turn generates more factor 12A and amplifies the pathway. Factor 12A also activates HK to release bradykinin, a pro-inflammatory mediator. This reaction is prevented by occurring in the plasma by the presence of the C1 inhibitor. Other exogenous artificial surfaces also can activate factor 12 and require anticoagulation therapy. More recently, it has been discovered that naturally occurring polyphosphates activated platelets, neutrophil extracellular traps, and cell membranes from bacteria can activate the intrinsic pathway and promote thrombin formation and thrombosis. Factor 11A also is able to activate coagulation factors 10, 5, and 8, and inhibit the anticoagulant tissue factor pathway inhibitor. Thus, factor 11 may have a role in thrombus formation and is thought to be a risk factor for DVT and ischemic strokes. The role of factor 12 and 11 in myocardial infarction, however, is not clear. In mice, factor 12 and factor 11 deficiency results in fragmented unstable thrombi, prone to embolization. Consequentially, the intrinsic pathway also may be essential for stabilization of thrombi and prevention of embolisms. It is important to recognize that factor 11A and 9A can be generated by routes independent of factor 12A. Thrombin activation of 11 and 9 is a feedback mechanism that amplifies the coagulation pathway and maintains the clot over time. Thus, the older intrinsic pathway nomenclature has fallen out of use to some extent because of preference for contact activation pathway nomenclature. Activated platelets are important participants in clotting. The phosphatidylserine-rich surface produced during activation provides a matrix on which several important complexes of clotting factor are formed. These include the intrinsic pathway 10-ACE complex and activated factors 8 and 9 that activates factor 10 and the prothrombinase complex that activates the prothrombin into thrombin. Thrombin then converts fibrinogen into fibrin, which polymerizes into a fibrin clot. Thrombin has broad reactivity in the inflammatory response. In addition to the production fibrin, thrombin is an activator of the coagulation proteins, platelets, endothelial cells, and monocytes. Vitamin K-dependent clotting factors include prothrombin, factor 7, 9, and 10, and the anticoagulation proteins. These proteins undergo vitamin K-dependent gamma carboxylation and glutamic acid residues that enables them to bind calcium and other divalent cations and participate in the clotting pathways. Vitamin K is antagonized by the anticoagulant drug warfarin. The control of homeostatic mechanism, therefore. The intact endothelium prevents thrombosis by inhibiting platelet aggregation, preventing coagulation activation and propagation, and enhancing fibrinolysis. Blood normally remains fluid despite the continual presence of clotting factors and platelets in the circulation. These major regulatory factors that control homeostasis reside where the greatest probability of clotting would occur, on the endothelial cell surface. The primary anticoagulant mechanism include thrombin inhibitors, tissue factor inhibitors, and mechanisms for degrading activated clotting factors. Antithrombotic mechanisms are listed in the following table. When the endothelium is damaged, it transitions to the homeostatic action described previously. So here we're taking a look at the antithrombotic mechanisms of the endothelial cells. Antithrombin III is a circulating plasma serine protease inhibitor produced by the liver. Specifically, it inhibits thrombin and several activating clotting factors. Your antithrombin III is produced by the liver and binds with heparan sulfate, found naturally in the surface of the endothelial cells, or with heparin administered clinically to prevent thrombosis. Heparin induces a change in the antithrombin III that greatly enhances its capacity to inhibit thrombin and other activated clotting factors. Under normal conditions, the presence of endothelial cells, heparan sulfate, and the available antithrombin III in the circulation cooperate to protect the vessel from the effects of spontaneous activated thrombin. Acquired antithrombin III deficiencies can result from infection with bacteria that produce antithrombin III inhibitors, sepsis, liver disease, and nephrotic syndrome, and lead to venous thrombosis and pulmonary emboli. Your tissue factor pathway inhibitor is produced by endothelial cells and platelets and is a primary inhibitor of the initiation of blood clotting. Tissue factor pathway inhibitors forms complexes with reversibly inhibits factor 10A. So the resultant TFPI-10A complex inhibits your TF and 7A complex and prothrombinase, and thus inhibits clotting. Although the majority of TFPI remains associated with endothelial or platelet surfaces, 20% circulates in plasma with lipoproteins. Heparin increases plasma levels of TFPI, which may contribute to heparin's antithrombotic effects. Thrombomodulin is a transmembrane thrombin-binding protein on the surface of endothelial cells and a receptor for thrombin. When thrombin binds to thrombomodulin, it no longer serves as a procoagulant and cannot activate platelets, converts fibrinogen to fibrin, and or amplify fibrin generation. Your protein C in the circulation binds to the thrombomodulin thrombin complex and is rapidly converted to activated protein C. Activated protein C in association with a cofactor, inactivates factor 5A and 8A, inhibits fibrin formation, and inhibits coagulation. Deficiencies of antithrombin III, protein C, or protein S are important inherited causes of hypercoagulation. Expression of thrombomodulin and endothelial cell protein C receptor is downregulated by cytokines and other products of inflammation. Decreased expression prevents protein C activation, thereby enhancing clot formation. Activated protein C inhibits the adhesion of neutrophils to the endothelium, but during inflammation, neutrophil elastase enzymatically removes thrombomodulin from the endothelial cell surface. Protein Z is a cofactor for the inhibition of the factor Xa through protein Z-dependent protease inhibitors. Lysis of blood clots. Concurrent with the activation of coagulation is the activation of pathways that limit the size of the clot and remove the clot after bleeding has ceased and repair has begun. The primary mechanism for lysis of blood clots is the fibrinolytic system, a cascade of serine proteases that produce plasmin. The plasmin degrades fibrin polymers in clots. The inactive precursor of plasmin is plasminogen, which is produced in the liver. Plasminogen activation may occur by several means, although the most physiologically important is by the action of tissue plasminogen activator, or TPA. Endothelial cells at the site of vascular injury express TPA, which is also a serine protease that reaches maximal enzymatic activity after binding to fibrin and proteolytically activates plasminogen to plasmin. Another activator of plasminogen is urokinase-like plasminogen activator. The UPA is a serine protease that can bind to a specifically cellular UPA receptor, causing activation of plasminogen and resulting in plasmin generation. This urokinase is the major activator of fibrinolysis in the extravascular or tissue compartment, whereas TPA is largely involved in intravascular fibrinolysis. Both TPA and UPA have been used clinically to treat diseases associated with a blood clot. So here you're taking a look at the fibrinolytic system. As with most components of inflammation, plasmin interacts greatly with other factors. In addition to activation by TPA and UPA, plasminogen is activated to plasmin by thrombin, fibrin, factor 12A, factor 11A, and kallikrein. Plasmin is a proteolytic to several substrates, including activation of collagenases, complement components C1 and C3, and factor 12, and cleaves fibrinonectin, fibrin, thrombospondin, laminin, and the von Willebrand factor. Plasmin activity is usually controlled directly by the serine protease inhibitor alpha-2 antiplasmin, or by inhibiting plasminogen activation by specific plasminogen activator inhibitors. Cross-linked fibrin is deposited in tissues around wounds, inflammatory sites, and tumors. Fibrin removal is an important biologic process for intravascular and extravascular spaces, with various controlling mechanisms that can lead to the abnormalities of fibrin accumulation and thrombotic events, and can be a structural barrier to tumor invasion. Products of fibrinolysis include fibrin degradation products. A major fibrin degradation product, or FDP, is D-dimer. D-dimer is a 2D domains from adjacent fibrin monomers that are cross-linked by factor 13A, and released as a result of enzymatic cleavage by plasmin. Measurement of levels of circulating D-dimer has been used for diagnosis of DVTs, PEs. Despite extensive literature, the diagnostic role of D-dimer is unclear because of the use of multiple D-dimer assays with different sensitivities and variabilities. So, clinical evaluation of a hematologic system. Tests of the bone marrow. The bone marrow is a soft, spongy tissue found within bones, especially the sternum, pelvis, and femur. Several abnormal conditions in the number of morphology of circulating blood cells or suspected infection of the marrow may justify further investigation of the bone marrow. Usually, bone marrow is aspirated from the sternum or pelvis using a needle. In children, a bone marrow aspirate can be obtained from the vertebra or the femur. The aspirate is examined microscopically and may be cultured if infected or infection is suspected. Microscopic evaluation may also include flow cytometry, chromosome analysis, or polymerase chain reaction related to the presence of atypical cells, the presence of atypical numbers of normal cells, and the absence of particular cell types. A normal bone marrow aspirate contains stromal cells, stem cells, and immature and mature forms of blood cells. The differential cell count of the bone marrow aspirate involves examining approximately 400 nucleated cells under oil immersion magnification and counting populations of cells differentiated on the basis of morphology. The relative number of each type of stem cell is expressed as a fraction of 400. So here you're taking a look at bone marrow samples. The following table here shows the differential cell count in bone marrow with age. Bone marrow iron stores, primarily in macrophages, can be examined using special stains for iron-containing granules. A direct measure of iron stores also can be obtained only from liver biopsy specimens. Although bone marrow is preferred because it is a safer procedure and because bone marrow is the immediate source of iron destined for erythrocyte production. Bone marrow aspiration is an important diagnostic test for severe central defects in hematopoiesis. Examination of the bone marrow is also useful to diagnose your B lymphocyte immune deficiencies, your non-malignant myeloproliferative disorders, and lymphoid monocytic malignancies, so your leukemias, myelomas, lymphomas. This test can also be used to monitor the effects of chemotherapy on malignancies that have invaded the bone marrow. A marrow aspirate that is richly cellular implies normal or increased hematopoiesis, but does not indicate whether marrow activity is effective. Results from bone marrow aspiration are sometimes limited because this technique disturbs the architecture of the marrow and only provides an analysis of the general cellularity of the marrow. On occasion, analysis of an aspirate may only suggest the presence of malignancy or a central defect in hematopoiesis, without being clearly confirmatory, or the sample may be inadequate for diagnosis of bone disorders or fibrosis. In these cases, the need for a bone marrow biopsy may be indicated. During a biopsy, a special needle is used to obtain a core or a cylindrical sample of bone and marrow in which the three-dimensional structure of the marrow is preserved. The biopsy specimen provides the most reliable and complete information about marrow cellularity. Obtaining a bone marrow biopsy is, however, usually more painful and extensive than aspiration. Therefore, biopsy is not performed unless insufficient information is obtained from aspiration. Which brings us to our blood tests. Blood tests provide information about the absolute and relative numbers of blood cells in a specimen of blood, as well as various structural and functional characteristics of the cells, and usually provide the initial justification for performing a bone marrow aspiration. Deviations from the normal differential distribution and presence of abnormal or immature cells can reflect disease, physiologic states, injury, dysfunctional in almost any part of the body. So blood tests that reflect chiefly hematologic disorders are listed below. So here in this table, you'll see all the blood tests for hematologic disorders. The ones that we would most likely expect to see would be the red cell count, your mean corpuscular volume, mean corpuscular hemoglobin, the mean corpuscular hemoglobin concentration, hemoglobin determination, hematocrit determination, reticulocyte count, erythrocyte osmotic fragility test, hemoglobin electrophoresis, a sickle cell test, glucose 6 phosphate dehydrogenase deficiency test, and then hemoglobin metabolisms. So your serum ferritin, your total iron binding capacity, transferrin saturation, porphyrin analysis, a direct antiglobulin test, antibody screening, and then your leukocytes, your differential white blood cell count, your neutrophil, lymphocyte, plasma cell, monocyte, eosinophil, basophil counts, and your platelet and clotting factors. So platelet count, bleeding time, your clot retraction time or test, platelet adhesion studies, platelet aggregation tests, your whole blood clotting time, circulating anticoagulants, and then the classics, the PTT and PT. And I think at this point there'll even be an anti-10A, thrombin time, fibrinogen assay, and fibrin-fibrinogen degradation products. Pediatrics and the hematologic system. Blood cell counts tend to... So, actually, let me go back to the top, only because I went through all this really quickly. So, make sure you hit pause if I'm ever going too fast. I'll try to stop where it makes sense. Okay, so talking about the pediatrics and the hematologic system. Blood cell counts tend to rise above adult levels at birth and then decline gradually throughout childhood. The following table lists normal ranges during infancy and the childhood. The immediate rise in values is the result of accelerated hematopoiesis during fetal life, increased number of cells that result from trauma of birth, and cutting of the umbilical cord. Average blood volume in the full-term neonate is about 85 milliliters per kilo of the body weight. The premature infant has a slightly larger blood volume of 90 ml per kilo of body weight, with the mean increasing to 150 ml per kilo. During the first few days after birth, in both full-term and premature infants, blood volume decreases during the first few months. Thereafter, the average blood volume is going to be around 75 to 77 ml per kilo. which is similar to that of older children and adults. The hypoxic intrauterine environment stimulates erythropoietin production in the fetus and accelerates fetal erythropoiesis, producing polycythemia of the newborn. After birth, the oxygen from the lungs saturates arterial blood, and more oxygen is delivered to the tissues. In response to the change from the placental to the pulmonary oxygen supply during the first few days of life, levels of erythropoietin at the rate of blood cell formation decrease. The very active rate of fetal erythropoiesis results in a large number of immature erythrocytes in the peripheral blood of full-term neonates. After birth, the number of reticulocytes decreases, about 50% every 12 hours, so that it is rare to find an elevated reticulocyte count after the first week of life. During this period of rapid growth, the rate of erythrocyte destruction is greater than that in later childhood and adulthood. In full-term infants, normal erythrocyte lifespan is 60 to 80 days. In premature infants, it may be as short as 20 to 30 days, and in children and adolescents, it is the same as adults, 100 to 120 days. The postnatal decrease in hemoglobin and hematocrit values is more marked in premature infants than in full-term infants. In the preschool and school-aged child, hemoglobin, hematocrit, and red blood cell counts gradually rise. Metabolic processes within the erythrocytes of neonates differ significantly from those of erythrocytes in the normal adult. Among other differences, the relatively young population of erythrocytes in the newborn consumes greater quantities of glucose than do erythrocytes in adults. Most coagulation factors in children reach adult levels by 6 months of age, and some not until adolescence. Children have quantitative and qualitative differences in clotting factors that result in decreased risk for thrombosis without increased risk for bleeding, because the reduction of procoagulant and anticoagulant proteins is balanced. Lab values for homeostasis are referenced by the testing lab. At birth, the lymphocyte count is high and continues to rise during the first year of life, and then steadily declines until lower adult values are reached. The lymphocytes of children also tend to have more cytoplasm and less compact nuclear chromatin than do the lymphocytes of adults. A possible explanation is that children tend to have more frequent viral infections, some of which are subclinical and are receiving vaccinations, both of which are associated with atypical lymphocytes. The neutrophil count, like the lymphocyte count, is high at birth and rises during the first days of life. After 2 weeks, the neutrophils count falls to within or below the normal adult range. By age 4, the neutrophil count is the same as that of an adult. The eosinophil count is high in the first year of life and higher in children than in teenagers or adults. Monocyte counts also are high in the first year of life, but then decrease to adult levels. Platelet counts in full-term neonates are comparable with platelet counts in adults and remain so throughout infancy and childhood. Which brings us to the aging and the hematologic system. Blood composition changes little with age. Erythrocyte lifespan in older adults is normal, although erythrocytes are replenished more slowly after bleeding, probably because of iron depletion. Values of total serum iron, total iron binding capacity, and intestinal iron absorption are all decreased in older adults. Iron deficiency is often responsible for the low hemoglobin levels noted in older adults. The plasma membranes of erythrocytes become increasingly fragile, presumably because of physical trauma inflicted during circulation. chronic inflammation, and the presence of chronic disease, including diabetes, heart disease, renal insufficiency, and Parkinson's disease, are associated with suppressed erythropoiesis and anemias occurring in older adults. Lymphocyte function decreases with age, causing changes in cellular immunity, with some decline in T cell function. The humoral immune system is less able to respond to antigenic challenges and to vaccination. No changes in platelet number or structure have been observed in elderly persons. Yet, platelet adhesiveness probably increases and may be related to oxidative stress. Levels of fibrinogen and factor V, 7, and 9, and also levels of the von Willebrand factor tend to be increased in older adults. In addition, thrombin generation and platelet activation are enhanced in older adults. Consequently, there are an increased incidence of venous thromboembolisms. So that concludes our chapter here today. I thank you as always for watching, and I'll see you at the next chapter. Till then, thank you and goodbye.